Good morning, everyone, and thank you so much, Jane, for the very kind introduction. I hope I can live up to those expectations. I want to thank all the organizers for inviting us and me here today to talk to you about our experience with Spark at Stanford. And it's now been 12 years since Daria Moshe-Rosen founded Spark at Stanford. And the goal was to advance discoveries from the academic research lab to help patients. So, interestingly, well, you'll see in a minute, the topic of the symposium, the Valley of Death, is something that we're trying to overcome. You all know that the pharmaceutical industry, big pharma, is involved in producing drugs, and they're very good at sales and marketing. But years ago, they basically shut down most of their basic research labs. They used to be developing new targets and drugs on their own, but they found that this was too expensive, and so they basically focused on the last phase of clinical trials and actually getting those drugs out there and selling them. On the other hand, biotech has sort of picked up some of that earlier phase research and runs preclinical studies. They try to optimize any compounds that come their way and run the early phase clinical trials. But there's still this gap between the early ideas that come out of academia and getting up to the point where somebody in biotech might be interested in trying to develop it. So there's a big responsibility now on all of you in academia to come up with these ideas, to do the basic research, and to try to feed the pipeline of therapeutics that will help patients. So how are you going to do that? There's still this gap, and that gap is what we call the Valley of Death in this situation, that most academic researchers don't know how to go from their basic research in the lab. What's the next step? How are they supposed to direct something to come up with a little pill at the end? So this is where Spark comes in to try to fill in this gap between your initial discovery and how to get it to the point to de-risk it enough that investors or other companies would be interested in partnering with you or licensing your technology. So the mission of Spark, as many of you know that are already participating here in Taiwan, is to form a partnership between the university and academic advisors, experts from, sorry, not academic advisors, but industrial advisors from biotech and pharma, who can help you figure out the process on how to move your discoveries forward to help patients. And so one of the major goals of our program is to educate faculty, grad students, postdocs in what this process is and how to go about it, and then how to advance these research discoveries along the path to FDA-approved treatments and drugs focused on unmet medical needs. So what I'd like to do is just tell you a little bit about what we do at Spark at Stanford, which is probably very similar to what you do here, and then give you some examples of a few Spark projects, discuss how we select these projects and what our process is for doing that and how to move them forward, and then give you some more examples at the end of successful projects from Stanford. So the first thing we do, which is similar to Spark Taiwan, is to provide seed funding for projects that have interesting basic research discoveries that have potential to move on to help patients. So we provide not very much money, $50,000 a year for up to 2 years, to try to advance these goals. And we try to leverage that funding as much as possible by bringing in other resources in the university and our advisors. So our advisors, as we have two here that you'll hear from later, are, I think, a big part of the secret sauce of making a successful project come out of Spark, they are experts in all aspects of drug development, and they can be medicinal chemists, they can be toxicologists, they can be regulatory affairs people, they can be entrepreneurs. So we have a vast range. We have over 100 advisors that we call on and who attend our meetings and adopt some of our projects to help them move forward. And basically with the help of these volunteers and our connections, we try to facilitate partnerships between industry and the researchers so that they can go the next step and develop their projects. So, oops, sorry if I'm blinding somebody with the laser. So what is our process at Stanford? You can see that we have all of our teams meet in the room at the same time. And I think this may be different from what goes on here in Spark at Taiwan. We have ongoing meetings every week, and teams to receive their funding are required to attend. So we have Wednesday evening meetings for 2 hours, and the meetings alternate between educational seminars by some of our advisors, as you'll hear, outside speakers, different topics about the drug development process. And then the alternating weeks, the teams give project updates on their progress since they came into the program or since the last time they spoke. So they, on average, give talks about once a quarter, each team. And so everybody is in the room together and learning from each other. So somebody's project may be at a stage that's well beyond yours, and so you're looking to where your project might be next year. Or it may be something that you went through last year, and you have advice to give them based on the problems that you overcame. So, as I said, we alternate these educational seminars with project updates, and all of these are under confidentiality agreements. So at Stanford, anybody who is part of Stanford in a closed meeting like this is under confidentiality and expected to not disclose any data or ideas that have come out of that meeting. In addition, all of our volunteer advisors sign non-disclosure agreements, confidentiality agreements, so that they are not supposed to discuss this outside the meeting either. So this really promotes a community of sharing ideas and learning from each other so that there's open exchanges. Graduate students and postdocs are on the same level as senior faculty, Nobel Prize winners. We actually have a couple Nobel Prize winners who are Sparkies. And all the advisors from industry, everybody can ask questions and provide comment at the same level. And as I said, people can share their lessons, both success and failure, as their projects progress. So I want to just give you first 2 short stories about projects that have come through Spark. And the first one is about a repurposed drug going to the clinic. So this project was based on a physician observation, and I have to give you kind of a backstory first about the molecule. This was a drug, sildenafil, that was originally developed by Big Pharma, and they had their own repurposing story in the development of this drug. So sildenafil was developed by Pfizer as an antihypertensive androgen drug, but it actually failed in the clinical trial. However, there was an interesting and unexpected side effect in that the men who were enrolled in the clinical trial for cardiac therapy didn't want to stop taking the drug. And that was because there was actually a sexual enhancement side effect with respect to this drug, and Pfizer ended up developing it as Viagra with a $2 billion market for erectile dysfunction. So in itself, this is a repurposing of a drug because they wanted to develop a drug for cardiac function, and they ended up with something totally different. But as a matter of fact, they did find that there was a benefit in a particular cardiac indication, pulmonary arterial hypertension. So that's related to now how this relates to Stanford's project. There was one of our dermatologists, Dr. Alfred Lane, who was treating a child for dermatological problems due to large lymphatic malformation that this child had, in addition to the fact that the child had pulmonary arterial hypertension. So because of these large lymphatic malformations that you can see here in the first panel, it was very deforming and stretching the skin, and there was a lot of problem that the dermatologist was treating. But over the course of weeks and months that the child was on sildenafil, he noticed the dramatic shrinkage of this growth, of this lymphatic malformation. And when the child actually went off of it for a short time, on the last panel on the bottom right, it started to grow back again. So it seemed to be due to the drug that this malformation was responding to. So Dr. Lane published this result, or actually he treated 3 other children, I think 2 other children, so 3 altogether on his own to see, and they also had positive effects. So he published this in New England Journal of Medicine, but then he didn't know what should he do with this. How could he test whether more kids were involved, how to get expand the study. So he came to Spark and asked for help, and we basically helped him design a small study at Stanford Hospital, treating more kids with these lymphatic malformations. And we also negotiated with Pfizer to donate the drug for his study. So for just over $50,000, he was able to run this pilot clinical trial, treating 7 kids, of which 6 had a very positive response. And this girl here, about 8 years old, you can see on the left side, she had quite a big growth on the side of her face. It was very deforming, affected her speech and eating. And on the right, you can see after 20 weeks of sildenafil, it really dramatically shrunk this lymphatic growth. And you can also see that there was a side effect, not attributable, to the drug, that she lost another tooth, but I think that was a regular developmental issue and not due to the drug. So Dr. Lane also published this result, and based on these findings, he was then able to leverage those to apply to the FDA and get a very large $1.6 million grant to conduct an even bigger clinical trial studying the effects of sildenafil. But so in the status of this clinical trial is that it's ongoing. They're enrolling patients at 3 sites and having positive effect. But sildenafil is cheap. It's generic, and it's off patent. So it's not a major commercial opportunity. But for Stanford Spark, this is not disincentive for accepting a project into the program, because what we are focused on is helping patients. Sometimes the only way to do that is to actually generate IP and develop a company. Sometimes when there is a drug that's already out there and it's cheap, but you can affect patients' health, you can just go ahead and develop that directly. And so that was what happened in this case. Because of the initial Spark support and his ability to publish and continue with clinical trials, it's affecting patients' lives throughout the world. People are using it for this indication. So the second story is about a diagnostic. Basically, Stanford Spark focuses on therapeutics, on drugs, and a small number of projects on diagnostics. As you'll hear from Fumi later, we don't do medical devices because we have the biodesign program at Stanford, although many of our other Spark programs, such as in Taiwan, also include medical devices in Spark. But this was a project that was selected in the first year of Spark, and it's focused on the unmet medical need of kidney transplants and rejection. So about 20% of kidney transplant recipients have an acute rejection episode within the first year of their surgery, and it's hard to detect this rejection process very much before it's too late to do anything about it, before you have organ failure. And so the gold standard for monitoring the status is to look at blood creatinine levels. And if they are rising, then it prompts a kidney biopsy, which is invasive. So basically, it's kind of a not optimal way of doing it. And you would like, and the creatinine levels themselves are not very specific in terms of prediction. So what this project that we Spark funded initially was looked at from the list of dockets from our Office of Technology Licensing, and they were invited to join the program at this time. And it was a collaboration between Dr. Mini Sarwal, who is a pediatric transplant surgeon, and Dr. Atul Butte, who is a bioinformatician. So together they analyzed public microarray data looking at differential expression of genes in tissues that were healthy versus tissues that were not healthy. And they were able to identify a panel of diagnostic proteins that were predictive of rejection in this particular case. So Dr. Sarwal went on to develop then a PCR-based diagnostic test. She called it the K-sort assay, the kidney solid organ response test. And they found that it was highly sensitive to even the very early stages of acute rejection. And it was at a stage where you could change the immunosuppression regimen and actually affect the outcome and prevent organ failure. Because of the results of these tests, Dr. Sarwal formed a startup company that she called Organ-Eye. She licensed her technology from Stanford to this company, and they then conducted an 8-center study to validate this K-sort test in over 500 patients. They found that they could actually detect this acute rejection over 3 months before the gold standard biopsy test. So after publishing these results, Organ-Eye was actually, acquired by Immucor in 2014. And in the meantime, since 2014, Dr. Sarwal has conducted several more clinical trials, which she published last year, and there's been over 1,000 patients now that have been analyzed by her test, and that found it to be very predictive. And you can see from the Immucor website that I captured last week, they are imminently getting ready to launch commercially the use of this K-sort test. Okay, so these are two examples of quite different types of projects that have been in Spark. How do we go about selecting projects? First of all, the common goal of projects in our portfolio is to rapidly move these basic research discoveries from the bench to the bedside. And as I just mentioned, our focus is on therapeutics, on drugs, and on diagnostics, not medical devices. So in the early years of Spark, like I mentioned for the K-sort test, Daria and Kevin basically went to OTL and looked through their unlicensed dockets to find projects that they thought could fit into the Spark model, and that they were novel and they were focused on an unmet medical need. And if so, then they were invited to apply to Spark if the project was far enough along that this small seed-level funding could help de-risk the project and move it forward enough within the 2 years of support that it had increased the likelihood that it could be licensed or obtain funding outside of Spark. So that was the early days, the first 3 or 4 years of how projects were selected. Since then, we run an annual call for proposals, and basically there are 2-page proposals that are submitted online, very focused on translational aspects of the basic research and why the investigator thinks that Spark support would help them and where it would take them. We also continue to interact with our Office of Technology Licensing, and they can suggest open dockets that they have and encourage the investigators to apply to Spark. So how do we review these projects and what criteria do we use to select them? We want to start with the end in mind. You know, basic researchers make discoveries and they usually use that discovery to continue down a path wherever it leads them. But translational research isn't like that. You have to have your basic discovery, you need that basic research, but you have to focus on the end. What does the patient need and how am I going to get there? So working backwards from the patient need, you have to define what clinical problem do you want to try to solve, what specific unmet medical need will your product address. Is the risk to benefit ratio acceptable? And that may depend on the actual therapeutic indication that you're addressing. Other issues that you can see there, will this be another Me Too drug like a statin, and the payers are going to say, why do we even need another statin? We're not going to pay you for this. And so that we would not be interested in something that's not a unique and novel approach. So in another sort of aspect of addressing the unmet clinical need is are these considerations that we are looking for projects where there are no appropriate therapies available, no effective therapies available. So an example is, even when there are no therapies available, is it a serious disease or is it not a serious disease? So clearly a Huntington's disease project where there is no therapy for Huntington's disease, it's a fatal condition, is much more interesting and appropriate for Stanford Spark to support than a cure for the common cold, which, you know, there is no treatment for the cold either, but it's really just an inconvenience and not a life-threatening situation. So these types of considerations are important in our review of the projects. Is there a possibility to not only treat the symptoms, but actually to arrest the disease progression or to reverse it? That would be something very important. Maybe the existing therapy works, but it has really bad side effects for the patients. And the new proposal would actually provide an alternative that doesn't have these detrimental effects. And finally, you know, maybe it's important for the patient to adhere to the therapy based on how they have to take the drug. If it's an IV infusion that's the current therapy, this is really kind of hard on the patient to do. And if you had something that could be given orally and it would be cheaper, this is a consideration that would be useful. So when we actually are reviewing projects, we usually are getting now 50 to 60 applications per year. We put together a selection committee of Spark advisors, some ex-Sparkies from Stanford who've had successful projects, and our Stanford team, our Spark team at Stanford. And everybody reads all the proposals and picks their top 10, makes their top 10 list. We compile all the results, and out of that we take the top 20 proposals. And then these top 20 are invited to pitch to the review panel. So the finalists, we basically, it's usually the first 2 weeks in December that we have 4 nights that we devote to the pitches. We have 5 projects a night, and they get a half an hour. They give their pitch and answer questions. And at the end of every night, the review panel ranks the ones that have presented that evening. Then after the last night, not only do rank the last 5, but then go back over all 20 and rank everybody, so that we end up with approximately 12 new projects per year that we have enough money to support. So not only is the ranking important, but also the funding source is important for our selection, because some of our funding comes from sources that are specific to certain indications, such as the Children's Health Research Institute focuses on projects, or wants to support projects related to children or maternal health. Other of our sources, such as the Leo Foundation, focuses on dermatological indications. And so sometimes a project that is maybe not in the top 12 might get moved up because of the funding source that's available for that project. So this is a compilation of how many projects have been supported by Stanford Spark over the 12 years, including our current number of projects. And I didn't discuss now, but partway through these years, we started accepting some projects off cycle if they've come to our attention, exciting results that we don't want to wait until till the next round. We can have them prepare a small proposal, pitch their result to a small, their project to a small number of reviewers, and give them less money to start with, and try to reach certain milestones in the interim before the next round of official applications happens. We call those seed projects, and then they can apply for full funding in that round. So 155 projects over 12 years. I was just talking to Jane yesterday, and this is not a very big number compared to Spark Taiwan. You guys have 5 universities participating though, and you are lucky to have significant government funding, is my understanding. So it's great that you are running through supporting lots of projects in the Spark program here in Taiwan. We wish we could come up with more money, and we're always looking for more funding sources, but it's a continuing struggle to put together enough funding to support the projects that we have. So the projects that we have selected over the years, we are basically agnostic in terms of what the therapeutic indication is. We support all kinds of therapeutic areas, chronic diseases, global health related projects, lots of pediatric projects. Some of those overlap with the chronic diseases, cancer in kids, that kind of thing. And here's another breakdown of how it looks in terms of our oncology supported projects are the highest number. And then you can see there's lots of others, neurological disease and many other indications. So then we come down to, while we're supporting all these projects, how successful are they? And how do we define success? This is the critical thing, right? So early on, Daria decided that we need to define success in terms of what we can control at Spark. Once some project moves out of the university as a startup company or as a licensed to Pfizer or somebody else, we don't have any control over that project at that point. So our criteria are based on what we can control at Stanford. And we define a successful Spark project as having entered a clinical study, clinical trial usually at Stanford, such as that first project that I talked to you about, Sildenafil. It's been licensed to an existing biotech or pharma company. This is perfectly great because they potentially have the resources to develop it further. Or it's been able to attract funding from venture capitalists or whoever that's led to the founding of a startup company. So based on these criteria, how are we doing? So here's our pie chart. We have about, at this point when we compiled this, we call graduated projects ones that we're not providing funding anymore, 2 to 3 years of funding, and that's all that we can afford to give to people. After that point, they're graduated. At that point, we have 88 projects. This was compiled last year. 25% have been licensed to a company and are in clinical testing now. About 19%, the red slice, is they have been licensed to a company and they're still in preclinical testing in animal models at that company. And these could be startups or existing companies. The green chart is pie slice is the clinical phase and not licensed, and again that would be clinical trials that we are conducting at Stanford generally. And then the 43% there are other, in the other category, that they either have failed proof of concept, or they've graduated, they're still pursuing the project, but they haven't yet been able to obtain funding. And so we still work with those teams to try to partner with them with people, with companies or funders. So another measure of success is how much funding can the investigator get after they are in Spark. And we have found that being in the Spark program with the educational and community discussions means that many of the investigators are much more successful at obtaining follow-on funding after they've been in Spark. They either have learned a lot more about the translational process, they have learned how to pitch, and they pitch in their grants, and this gives them a leg up in the funding agencies and getting them more funding to support their basic and translational research goals. So it's about fivefold, and I think this is actually a minimum estimate because we're not that good at tracking all the funding sources that people get after Spark, but it's at least a fivefold increase in additional funding. But we come back to the failures. You know, why do projects fail? So the most common reason is the failure of the team to execute. So when they come into Spark, they define milestones and objectives and a timeline. And unfortunately, even despite all the education and the meetings, sometimes people are really still basic scientists at heart and they don't actually adopt the translational model very well, so that they make a new discovery and thought, wow, that's cool, I'm just going to keep going down this path instead of the path that's over here to actually go to the drug. And so, you know, sometimes you just can't teach a dog new tricks, but this is actually a relatively small number. It's the most that we have, but a small number, and we're always working to try to fix that problem. Other ones are kind of more hard to prevent. If you're developing a small molecule and you can't come up with something that hits your target that's not toxic and that doesn't have other kinds of problems, sometimes you just cannot find a good development candidate for based on your initial basic research discovery or target that you're trying to address, or the clinical model that you're trying to solve, to provide a therapy for. Sometimes you had a hypothesis and it just was wrong, okay? That is, I mean, we try to weed those out in the review process, but you know, sometimes you're not 100% right on your hypotheses as a scientist. That's why you're doing science. You don't know the answer. So sometimes it just fails, the preclinical proof of concept. So those are the most common reasons. There's a couple other less common reasons that we are working to address. But basically, we are trying, as I mentioned in that purple part of the pie chart, we have these failures and they're sort of out of the program, but there's still a significant number of projects that are not being funded, but that we are still supporting in business development efforts. So we coordinate with our Office of Technology Licensing to try to generate interest in investors or companies in our particular projects. And you might say, well, why are we doing that at Spark? That's the job of OTL. The thing is that OTL has dockets from the whole university. So they cannot dive down and know the nitty-gritty details about every single one of those projects or spend the time going out trying to sell them to everybody. They have a database and they put the information on the web, non-confidential, but basically we at Spark are much more intimately engaged in our projects and project management. And so we focus on the projects from the very early stages of coming into Spark all the way out to trying to graduate to get them partnered. And so, excuse me, even at the very early stages, if we have some new oncology projects and we're talking to companies and we say, hey, we have a project that we just took on, you should keep in touch, so as this project progresses, you may want to take a look more closely. So we collaborate with the project PIs to develop non-confidential information that we can pass along to interested parties. And we try to develop connections for them. We have all our, as I said, over 100 advisors in all phases of drug discovery and entrepreneurship and starting companies and actually being CEOs of companies. And so we connect them together, also potential funding groups. We attend professional meetings such as bio, and as Jane mentioned, we also highlight some of our Spark projects at the BioInvestors Forum in the fall, and you've had several teams from Spark Taiwan who've come there to pitch their projects, and we then do the same thing with our internal projects. So when we have made connections with potential investors or companies interested in licensing the IP, and we've gone past the initial stages of discussion, and the potential partner is very interested, then we hand it off to our Office of Technology Licensing, because they're the ones who actually craft the licensing agreement and the funding terms and all of that. So at that point, we are not involved other than just trying to track the results of the negotiations. So basically, that's an overview of how we select projects, how we support them, and I will just give you a few more examples of Spark cases that might be informative in the types of projects that we pick and where they go. So you recall the first story that I told you about sildenafil and lymphatic malformations. That was a drug repurposing project, as I mentioned. So what do I actually mean by that? And I'm telling you this because the next couple of projects are also going to be drug repurposing that I'm going to be telling you. This is a drug with prior experience in humans, and it can be either an approved drug, it can be something that a company is currently developing, it could be something they did develop and went through clinical trials but failed. And as long as it's not failed because it's totally toxic, it could still be of interest in a different therapeutic indication. So why do we care about this? Because generally you would expect that somebody else owns the IP, and why would you want to do something with a repurposed drug? Well, the first example was that you can help patients with the kids with lymphatic malformations. But you also can develop IP on these molecules, even though you weren't the one who actually developed it initially. And the reason for wanting to do it is that it's been in humans already, and so it's much faster to get to the approval state. You don't have to go through all the preliminary toxicology that you'll hear more about later. You have already safety studies that have been done. So you can sort of leapfrog many of the steps on that development pathway to get further along and get it to be more attractive to partners. So you have also the potential, as we saw, to change clinical practice, and as I started to say, you can potentially develop IP by developing a novel formulation or method of dosing, or you can file IP for method of use for your novel indication. So just one more word about the background to this. Even though it's an approved drug, and if it is an approved drug, a doctor could prescribe it to anybody for anything that he or she wants. But if you want to do research and show its effectiveness other than just a one-off case by case, you have to go to the FDA and explain what you want to do, and potentially file an IND, investigational new drug application, if you want to change the label of the drug, and if it potentially is being used in a different population, like kids instead of adults, where the risk is higher for that population. Maybe it's a different dose. So you most likely, even though it's a repurposing project, will still need to interact with the FDA to get approval to do a clinical study. So the first other example that I'm going to tell you about is another one from the first year of Spark. And Dr. Sharit Anand was a postdoc with Dr. Jeffrey Glenn at Stanford, and they were working on anti-hepatitis therapy, hepatitis C. And they did a screen that identified clemizole, which was an antihistamine approved in Europe, as an inhibitor of the replication of hepatitis C virus. So they published these results, and then they came to Spark, and we worked with them to actually get the supply of the drug from Europe, to run a small clinical trial at Stanford showing the effectiveness of the drug, and identified a venture capitalist to invest in a startup company based on this initial discovery. This was called Eiger Biopharmaceuticals. They are currently developing a clinical pipeline for orphan diseases, and in fact, they have licensed IP from 3 other Spark projects at Stanford. And in 2016, they did a merger with Celadon. Actually, I don't know how the negotiations worked, but they basically acquired their public status, their status as a public company that's listed on NASDAQ. So Eiger is now a publicly traded company in the United States. Based on this, so as I mentioned, Dr. Glenn was the founder of Eiger. Dr. Ainov, who developed the first screen in the identified chromosome, was then able to get a faculty position at Stanford and set up her own lab. And she continued working on antiviral therapeutics. Her idea was that, well, you don't want to stick with this old approach of every kind of new virus emerges that you need to develop a new drug. They all infect cells, so isn't there some way that we can figure out how they're utilizing the cellular machinery and attack that process that would be common to multiple viruses? So instead of this one drug, one bug approach, she identified 2 proteins within the cell that actually facilitated the life cycle of multiple types of viruses. And in order to inhibit this process, she found there were 2 anti-cancer drugs that were approved that in combination inhibited the viral life cycle. And so she developed these antiviral therapies, and they were effective in animal models of multiple viral diseases. You notice that Ebola is one of those, and this research was done at the time of the outbreak of Ebola in Africa. And with the help of Spark, she developed a clinical protocol that had to go through the CDC and the WHO and be sort of get into the pipeline of trying to treat people in Africa. And luckily for those countries and people, the Ebola epidemic by that time diminished, and so there weren't enough people getting infected that they would actually apply the protocol. But now they have in hand an approved WHO protocol that if an unfortunate event would happen, that Ebola emerges again, they would be able to test this drug combination. So again, notice these are 2 anti-cancer drugs in combination, and so based on that IP of this antiviral combination therapy, it was licensed to Globovir Biosciences, which was a startup not founded by Sharif, but it was a local startup. And they're planning clinical trials studying treating dengue virus in Colombia. And in addition, as another example of getting follow-on funding, Sharif was able to get a large Department of Defense grant to continue developing antiviral therapies, and she's working to develop novel derivatives of these anti-cancer drugs, which have some toxicity issues. Another example of an early Spark project was carried out by Dr. Garner. He is a neuroscientist who was studying cognitive disorders and Down syndrome in particular. He found that if you inhibited a certain neurological pathway, the GABA-A pathway, that you were able to normalize memory, and he had a mouse model of Down syndrome in mice, that treatment with this repurposed drug, aniline tetrazole, was able to normalize the cognitive function in particular mouse models. Came to Spark, he published these results again, came to Spark. We helped him to basically develop to test the doses that don't that are effective but don't have the side effects that this particular drug has in terms of causing seizures. Also found, interestingly, the timing of the treatment was very important, that the circadian rhythm is very important in consolidating your short-term memory into long-term memory. So the point at which you give the drug, the timing of the day, was very important for its effectiveness. We also helped him identify a CEO, and he started a startup company, Balance Therapeutics. They raised Series A and B funding and did a clinical trial with positive results in Australia. So they are currently continuing to investigate this molecule and others that are involved in cognitive function and applying them not only to Down syndrome, but also to aging, cognitive deficits, Alzheimer's disease, and those types of problems. The other just point I want to make about this project is early on, people thought that Craig was crazy to try to work on Down syndrome because nobody thought this was a viable therapeutic indication, that you could do anything about it, right? He tried to sell, to get pharma companies to license his technology. He didn't want to start a company, but nobody was interested. They just said, forget it, it's not going to work. So the fact that it is working is great, but also once he published and showed results and actually talked about his work to these different companies, basically it stimulated the whole field, so that now there are drug companies and biotech companies, other companies are investing in trying to address these cognitive disorders, even such as Down syndrome. And so he's very proud of the fact that even just his research has caused other people, his competitors, to try to develop more therapies. So Dr. Speaker Cutter at Stanford is a clinician scientist who studies indication that we heard about before, pulmonary arterial hypertension, briefly, and you saw that that they were using sildenafil to treat that underlying condition. But she also found that there was a subset of patients who were not responding, and she found that they had a mutation in this particular protein, and the signaling in that pathway was dysfunctional. She did a screen and found again that a high-throughput screen of FDA-approved drugs, and found that a drug called tacrolimus, which is used as an immunosuppressive agent for transplant patients, was actually extremely effective in treating this disorder. So she came to Spark, and Spark helped again with clinical trial design and various other aspects of this and obtaining the drug, and helped to set up the clinical trial at Stanford. So she found, she ran a clinical study, she found that it was effective in some patients, and in addition, she had 3 end-stage pulmonary arterial hypertension patients who were too sick to actually qualify for the clinical trial, and she got approval for treating them with compassionate use. And they were basically on the lung transplant list waiting to see if they could have a donor. And after treatment with tacrolimus, they basically improved their condition so much that they were taken off of the lung transplant list. So basically, you have to find the subset of patients that have these particular mutations in that gene in order to test whether this drug is going to be effective for them. She licensed the IP to Celton Pharma, and eventually it was the whole idea was granted orphan drug status, both in the United States and the EU, and the license has been transferred and is now held by Vivus, who is trying to develop it further. So I've been focusing a lot on repurposing because, as I mentioned, that hopefully, as you can see, it's a faster path to market, to patients, and sometimes to market as well. But we don't only focus on those. We do have novel molecules that we are developing. So just quickly run through a couple other cases. Peter Santamaria is an ear, nose, and throat surgeon from Australia. And he actually came to Stanford, I think, originally as a biodesign fellow, and then became interested in the SPARK program because of his observations about kids in Australia in the outback. He had clinics in the outback treating kids who had very frequent ear infections, and many times they had perforated eardrums. But he wasn't able to do surgery in those clinics. He didn't have the facilities. And so he was trying to figure out, well, there must be some way that we could try to heal these eardrums faster, prevent these recurrent infections, and do something for these kids. So his idea was to use heparin-derived epidermal growth factor in a formulation that you could just put in the ear so that it coated the eardrum and accelerated the healing of the perforation. And so this idea he published in a paper, filed IP on it. SPARK linked him up with one of our advisors, Ben McGraw, who became the CEO of the startup company that they founded called Oration. And they continued to try to raise money to expand clinical trials on this biologic molecule. And in fact, in 2017, they had a deal with Astellas, who licensed the IP and is developing the new drug. So you may have noticed, and I think you may be getting some copies of these slides, that I have some YouTube video links on them, and these are from our 10th anniversary symposium that we had at Stanford 2 years ago. And some of these videos are really great to watch. And Teresa is an inspiring video if you have time to look at it. She is a neurosurgeon who came to Stanford for her residency and fellowship. And she was also interested in basic research, research related to her interests on childhood brain cancer. So medulloblastoma is the very most common childhood brain cancer, and many of them are driven by problems, mutations in the hedgehog pathway. And she found a kinase that is critical for regulating this pathway at many steps. And she developed a novel screen and showed in the animal models over there that the drug can be very effective in stopping and reversing the disease. So the problem is that the common treatment, the current treatment is surgery and radiation, which in and of itself, if the cancer doesn't recur, can be very devastating to the child's development. So it would be great to try to get past that kind of treatment. So she, as I said, was a clinician. She was doing research in the lab, and she is the first to say that she had no idea that she herself had any capability of developing a drug for patients. She didn't have any idea what to do. So she came to SPARK, and we helped her complete the preclinical work, clinical trial design, approached the company. This is not an approved drug, but it is in clinical testing by Senwa, and persuaded them to support her study, and they are now part of the clinical trial process. And the clinical trial has been accepted into the queue in the Pediatric Brain Tumor Consortium in the United States, and they're hoping to start the trial this summer. So finally, I want to use an example from Daria's lab in Chehong. Chen is here today. I thank him for the slides. And I just want to quickly use this as an example again of the timeline of how long it takes to develop projects to a certain stage. So Daria and Chehong have been doing research on aldehyde dehydrogenase 2. Many of you may have heard Chehong speak about this before. This enzyme basically detoxifies aldehydes, and it's very important in everyone for all kinds of toxic molecules that we ingest in one way or another. But they also found that there's a particular mutation in East Asians which really causes a deficiency in the enzyme activity. And that leads to the Asian flushing syndrome when people are drinking alcohol, but this is more than just a cosmetic inconvenience. The mutation actually increases the risk of head and neck and esophageal cancer as well. So it's really an important clinical need. So they used high-throughput screening to identify novel molecules to activate the enzyme. So many times targets you're trying to inhibit enzymes, but in this case they were looking for something to activate the deficient enzyme. And I don't want to go into all the details of the development, but you can see that they started, the project was accepted into Spark in 2007, supported for 2 years. At the end of 2 years, they had made a lot of progress, identified other molecules, published the results, and then started a company that they called Aldea. They spent the next 2 years progressing the science, identifying other molecules in the pipeline, what the therapeutic indications were they going to go for besides this particular mutation in that population. And then at the end of those 2 years, they were funded by US venture capitalists, raised Series A and B funding, finished their preclinical studies, and did IND filing in a phase 1 study by 2014. Okay, at that point, I don't want to go into the details. You can talk to Chehong about the issues that went between what happened with Aldea and basically transferring the IP to a new biotech, Aviv. And then another 2 years passed with ongoing work and reaching out to potential funders, and they have been licensed. The IP now for this project has been licensed to 4C Pharmaceuticals, a Taiwanese company, and they are basically in preparation for a phase 1 study on peripheral artery disease. So it's a long process. That's bottom line. So I've mentioned some of these startups that came out. I don't want to expect you to read all these. If you have, hopefully we'll get copies of the slides. You can see what all of these different startups are focused on. Another batch here. And then in addition, as I mentioned, they don't always have startup companies. We have projects that are licensed or optioned to existing companies such as these. So basically, I just try to give an overview of our experience at Spark and some of the projects and cases that we have supported over time and their outcomes. But I want to point out that Taiwan was basically our first international Spark partner. And we have 5 great anchor universities here engaged in the process. And because of your example, it is clear to many other institutions and countries that it's not a unique Silicon Valley program that's at Stanford, that it can be replicated many places, and there are now programs all over the world in all countries and continents, not all countries, all continents except Antarctica, are either starting or have started Spark programs. So what we're hoping is, as a network of translational scientists without borders, that we can collaborate with each other and build synergies so that you can develop your own institutional and local biotech or therapeutic indications, but that together we can address bigger problems as a network and have synergies that will affect even bigger areas. So finally, just why should we keep sparking? I think I tried to tell you that it's important to educate the faculty, postdocs, and grad students, and especially the latter, because most students and postdocs are not going to get academic jobs. There aren't that many academic jobs, and so they're going to end up in jobs in industry, and this training will be very valuable for them to know what they're getting into and to contribute quickly when they join their new position. Also, as we've seen, it can trigger biotech and development of therapeutic pipelines for companies in your local area. But really the most important thing, I think, is that we want to translate this amazing science that's happening in academia that could die on the vine if it's just going through the normal academic process. And instead we wanted to flower and become products that benefit patients. And we feel that since so much of our basic research is funded by government, and that is by the public's tax dollars, that we have a social responsibility to try to take these discoveries and benefit patients and the public. So I thank you for your attention, and I'll be happy to answer any questions.
这是很好的机会，大家可以用力的问哈，我们刚好还有一点时间。好。
Sorry, in this journey cruising through the valley of faith, what kind of role the creativity can play? Because in many of your cases, I found that these scientists and the researchers, they possibly have some fortunate and surprise finding, benefit finding, so that they can develop a new drug or so to really go through the spark and get funding and something like that, right? But is there anything that we can do if we actively participate in the process, not just by chance or our fortune, to find the new applications of the drug?
I'm not exactly sure. I think that clearly the basis for all of this is still supporting basic research. So basic research is the foundation for everything, and that's a combination of persistence and luck and funding and everything. So I think the important thing is to keep doing your basic research, but have the end in mind, that not only do you want to discover mechanisms of biology and how does it work, but that you want to benefit patients in the long run. And so if you make a discovery, well, is this something that could be useful, and then how do I do it? At Stanford, the other thing I would say is that in all of our educational seminars and in the project updates, students who aren't in the program and postdocs as part of Stanford can participate and come to these seminars and discussions. And so even if you're not at the stage yet where your project would be at the level that could be helped by Spark, you can still learn what it takes and where the process is going. So does that answer your question?
I have a question regarding the failure rate at Stanford. It seems to me it's very low, right? I mean, according to this book, 11% of the academia invention cannot be reproduced by engine, okay? And for example, in Taiwan, it's very difficult to license our products to companies. Therefore, we need to invest a lot of money to sort of push those products into clinical trials. But the problem is, this is my idea, okay? We are thinking to use the third party, like CRO, to sort of confirm the efficacy of the products. So I just wonder, how do you select your project with such low, failure rate? Thank you.
That's a very good question. I think I know the studies that you're referring to, and a lot of the issues are some of the things that we try to train in the Spark process, in that as Daria found when she was, even before she started Spark, she had made discoveries and she couldn't get anybody to invest because they said, well, academic research isn't reproducible. And so she even went around and she had other labs do the research, reproduce her results in other people's labs at other institutions. So I think it's a very difficult question, and certainly we don't have the money in most cases, certainly for novel molecules, to take things to the stage of a clinical trial at Spark. We have, the only ones we can do are ones that are repurposed drugs that we can take directly in. So I think it is still this valley of death problem about how to get reproducible results, to convince the industry or VCs that your results are reliable. I think having advisors such as Mike and others, and medicinal chemists who can help define what is industry looking for, what tests do you need to show that that this molecule has potential to progress. I think potentially using CROs would be a great idea if there's funding to do that. I think coming back is the funding is a big issue. So they're not cheap generally, but I think that would be a great idea.
Right. Yeah, Nancy, very nice talk. I got a question regarding the licensing policy in the SPARC, in the Stanford. Yeah, can you give more details for how the licensing policy in your institute? Because in here, I've experienced something of bring out a company, their interest, but the problem, their licensing process sometimes stop there, right? Mm-hmm. My first question. My second question regarding, since it's an international SPARC program, right? Do you adapt any international project to the Stanford program? Yeah. Maybe coached by Stanford program. Thank you.
You could ask Jane about one of those projects that we're trying to co-develop. But to your first question about the licensing, I don't know all the details since that's from our OTL office that does that. But so what can I actually say to address that question? I mean, a lot of our projects, as I mentioned in that purple side, are looking for somebody to license their IP. So we try to help with that part. Basically, the process is for investigators at Stanford, if they make a discovery that they think has potential to be patentable, they file an invention disclosure with our Office of Technology Licensing. And that is just sort of putting a stake in the ground as here's when I made my discovery and here's the information I have at this point. And they work with OTL to decide when to actually file a patent. And OTL doesn't want to file a patent because they pay for it, unless they think it's a high probability of it's going to be able to be licensed out, either to a startup company or to an existing company. And so it's kind of based on the feedback that they get and how much interest there is in the project by potential partners that they decide whether to, and at SPARC, our advice about what we think the potential market is or the potential to actually be successfully developed. Whether they will pay the patent costs up front and go ahead and file, or how long they will wait. Sort of when people are getting ready to publish, then they want to be sure that they will file their patent application before they publish the results. So at a point where, I think I probably don't have that much more information to share about that process. And we do have some international collaborations. Jane, when she came to Stanford for business development purposes, and is now conducting an autism study with one of Stanford's professors, a clinical trial. We also are we're trying to promote collaborations. So I want to be clear that we have not gone out and founded SPARC at all these places. Basically, people hear about the program, feel the need for training and helping their projects achieve success in licensing or starting companies, and they approach us saying, we want to start SPARC, can you help us? What do you need to do to start SPARC? And we sort of distribute a bunch of materials, and a lot of times they come to Stanford and come to one of our meetings and see how it runs, and we just sort of provide advice. But all of these different places are running their own programs. We're not running them, and many times they are doing somewhat different approaches than we do, but they all have the same common goals of trying to advance therapies to patients in the fastest way possible.
So I guess I do have a seed fund from the Stanford Spark through the Spark collaboration. So I was there last year for 40 days, supported by our government. And I think one of the great benefits that they do have, like, so just think about this. If you have your project being held by like 1/3 of people here who are industrial like advisors, they can help you for free and help you to develop and help you to reshape your program. Your program can move like in a really super fast speed that, you know, along the path. And a lot of things that we're not clear, they do have that ability to help with you. And also sometimes even finding the new potential better indication. So my findings with the autism was through that type of the, you know, advisory help from the Stanford. So we do still have the slot opportunity to go there for 40 days, and the Spark program, Spark office will actually help to support your expenses there. So talk to Spark office here, and you know, you might be the one to go next. Any other question? If not, then I have a question. So when I was there, something like a couple things that really surprised me. Although, you know, I knew that I'll be seeing a lot of advisors, which are extremely helpful. Mike was there, and we really get a lot of help from Mike. Couple things. I realized the PI for the Spark project that you support, a lot of them are PhD students, postdocs.
Well, there are a few students, but some postdocs. But the faculty have to be on board because the money can't go to the postdoc or the grad student. It has to be administered, granted to the faculty member. But yes, many times it's actually the driving force is the student or postdoc.
Okay, and the third surprising factor is that I saw a lot of naive MD. Excuse me for using that term, but you know, I saw a lot of MD with, you know, it's very easy for them to identify the medical needs, but they had no clue how to develop a drug. Right. So how does Stanford Spark manage to educate them? Because those are the hardest group to develop a drug, you know, compared to a medicinal chemist who already has some compounds and just looking for indications.
I don't think we do that much different for the MDs versus the PhDs or the students, except that I mean, it's all individual cases because every project is so different. Every project has a different need, is at a different stage, and so since we have such a large group of advisors to draw on, and we have real expertise in working with the FDA within the Spark directors, and you know, they know what is necessary to move things forward. They have lots of contacts with CROs and companies that can help with the process. So I think that it's individually, but Teresa that I mentioned with the medulloblastoma is a prime example of an MD who was interested, made a discovery, didn't have a clue what to do with it, and it was really the coaching and very individualized by Kevin that helped her know where to go. And from presenting to the Spark meetings and getting feedback from all the advisors, and you know, making the connections, that has helped her progress her project. So a lot of times the MDs will be presenting to the group like this, and advisors will say, you know, that's crazy, you know, they're usually not too rude, but they will basically make suggestions that what their original thinking is not that accurate, or that they need to do this step before that. So I think, as I was trying to emphasize, it's the community of engagement by all the Sparkies and the advisors and the Spark directors and staff that help lead each individual project in the direction toward helping patients. And I think there's not a general answer except that it takes a village to advance everything.
So one of the great benefits for being in the Spark is obviously the huge group of the advisors. So I was there, every week, and then I remember I wrote down on my Facebook and say that, I wish I can audio recording the whole thing. If I write it down, the debating between different advisors and the suggestion to the project, and that would be a great teaching material for the drug discovery. So I talked to a lot of them, and it seems that they've been with Spark for a very long time. So how do you keep, getting the new advisors or keep your advisors? I mean, those are all, I know they don't get paid except the dinner. Every gathering, they eat very well, It's very important. So how do you manage to do that?
I think you have two advisors here who could address that.
So maybe I'm one of those folks. I've been involved with the Spark program for a number of years now. But I don't think I'm much different than the rest of the advisors. We find it rewarding, our participation is rewarding for ourselves. So there's some inherent benefit, right, to the individual. As well as, I mean, it's an academic setting. I mean, there are a number of students involved with these projects, so it has its own type of a reward system built into it in that regard. And plus, I love being able to park like 5 miles from Stanford and have to walk all the way. I'm kidding. Parking's always an issue. But I think it's just a very rewarding setting for both parties. The folks that are directly associated with Stanford, and then the advisors, mentors that are participating.
Right, I would add, it's just fun. It's, you get to go to these meetings and listen to these really interesting, clever scientists who have come up with new approaches and new ways of attacking thorny problems, and it's rewarding to sort of be able to help any way you can, but just being exposed to that environment and listening to the other mentors with their comments, you learn a lot as an advisor, just listening to the rest of them provide their comments and input and learn about really interesting science at the same time. So it's just fun.
Well, I wish we can one day join any review session of our own project and think, oh, hey, this is fun. This is not like my blood pressure is very high. I need a new drug.